Welcome back. So for this lecture, we're going to talk about blood. We're going to go over the characteristics and functions of blood. So I want you to know just the basics for that portion, the components of blood, and then characteristics of each of them, the red blood cells and white blood cells, and then hemostasis, which is blood clotting, and then the blood typing. Blood is connective tissue. So in anatomy, when you covered the various connective tissues, blood was one of them. It is unique because the formed elements are actually the cells. So your red blood cells, white blood cells, and platelets are the formed elements, and the plasma is your liquid matrix. So it's unique in the fact that the plasma is liquid matrix. In most connective tissues, a matrix is solid. So 55% of the blood is plasma, 45% are the formed elements, the red blood cells, white blood cells, and platelets. Formed elements develop via hematopoiesis in red bone marrow. So there's stem cells in the red bone marrow, and they give rise to your red blood cells, white blood cells, and platelets. Average volume of blood, 5 to 6 liters for men, 4 to 5 liters for women. It's based off of body size. Men are going to have more red blood cells because of testosterone. Testosterone increases metabolism, and because men have an increased metabolism, they have an increased demand for oxygen. So their red blood cell indices are going to be slightly higher. That means red blood cell count, hemoglobin concentration, and hematocrit. pH, I want you to memorize this. So a lot of times when you know physiological ranges, they're slightly different depending on the book. pH is not one of those. So pH for blood is 7.35 to 7.45. Don't give me 7.4. Don't give me 7.3. The pH is 7.35 to 7.45, okay? That's really important because when you shift the pH, let's talk about pH for a second. So what's neutral? 7. As we move up in the scale, is that acidic or basic? That is basic. So as we're moving closer to 14, you add OH, right? Or you take away H+. As you move down the scale towards 0, you're making it more acidic. So you're adding more H+ or taking away OH-. And it's a logarithmic scale. So that means from pH 7 to 6, you have 10 times the amount of H+. From 6 to 5, you have 10 times the amount of H+. And if this is confusing to you, you should look over pH. pH is really important to know. Don't tell me that pH is going to increase and it's going to be more acid. That's not true. Increase in pH would make it on this end and it's going to be more basic, right? So as you shift the pH to a more basic, as you shift the pH of the blood to more basic or more acidic, that's going to change how the hemoglobin fold, right? Remember, they're proteins, and when you shift the temperature, the pH of proteins, that's going to affect their folding. And when these guys start to unravel, they're not going to hold oxygen, and that's going to be a huge problem for you, okay? So I want you to memorize that range. Your formed elements, the proper terms, erythrocytes are red blood cells, leukocytes are white blood cells, and thrombocytes are platelets. Here's a scanning electron micrograph, magnified 2,600 times the original size. It was in black and white or grayscale, but they colored them in. So these guys are the red blood cells. Notice that they're biconcave discs, right? These are white blood cells. Some of them are about the same size as a red blood cell, and some of them are way bigger. What are these little raisin-looking guys? Good, they're platelets, okay? Take a look at a red blood cell. 7.5 micrometers and 2 micrometers deep, right? 7.5 micrometers in length and then 2 deep. Pretty small, huh? What's the point of blood, or why do we need blood? Transports gases, nutrients, and hormones. That's pretty obvious, right? We all know that. You need to get oxygen to the tissues and CO2 out. Regulates pH with kidney and lungs. Electrolyte balance and interstitial fluid with your kidneys. Defends against pathogens and toxins, right? Your immune system. prevents fluid loss from bleeding, hemostasis, be careful, hemostasis, so this would be blood clotting, and aids in body temperature regulation, which you guys are already pros at, right? Here's a component of blood. So take a look. And this is, make sure you know your units, right? Percentage by body weight. So blood is 8% by body weight. If we took blood and we stuck it in a centrifuge, which spins it at a really high revolutions per minute, right? RPM, the red blood cells would be heavy, so they would sink to the bottom. Then we would have the buffy coat and the plasma. And so when we look at blood, and it's separated, the formed elements make up 45% of the blood, and then plasma makes up 55%. All right, take a look at formed elements. What are they? Red blood cells make up a huge amount of that, then white blood cells and platelets. Take a look at the number. So formed elements, number per cubic millimeter. So we have 4.2 to 6.2 million red blood cells per cubic millimeter. So think of a millimeter as a square, right? Cubed. 4.2 to 6.2 million. On average, it's about 5 million red blood cells per cubic millimeter. That's massive. Take a look at your white blood cells, 5 to 9,000 per cubic millimeter, and platelets, about 250 to 400,000 per cubic millimeter. When we talk about white blood cells, there are 5 different types of white blood cells: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And we'll talk about those about halfway through the lecture. Neutrophils are the most abundant, about 40 to 70%. You'll notice that these numbers are slightly different from textbooks and slightly different from your lab. That's okay. Pick one number that makes sense to you or that you can memorize. Or pick the lowest number, and that way you know anything below that is deficient, right, in some way. So neutrophils, about 70%. Lymphocytes, 20%, right? Monocytes, 3, eosinophils, 2, basophils, about 1, right? So if we did a white blood cell count, if we counted 100 white blood cells, we may see a basophil, we may see 1 to 2 eosinophils, we should see about 3 to 5 monocytes, about 20 lymphocytes, and about 60 to 70 neutrophils. So in a white blood cell count, as you would do in lab, which we're kind of going to modify for this lab, most of the white blood cells that you would see would be neutrophils. And so I tell students in lab when we're doing face-to-face that if you're not sure, it's probably a neutrophil, right? How do we remember the frequency? Never let monkeys eat bananas. Never let monkeys eat bananas, okay? So most abundant to least abundance. I want you to know these numbers, right? You should memorize all of these numbers so that you have them. So that would be a good use of flashcards. Normally, I don't recommend flashcards because you want to be writing and drawing and processing and synthesizing the information. But that would be a good use of flashcards. Okay, so let's look at the buffy coat. This buffy coat, the white layer, is made up of white blood cells and platelets. And then plasma is 55% of your blood. Most of it's water, right? 91%. There are some solutes in there, like ions. Can you give me an example of an ion that is found in plasma? Good. Sodium, which is a +1. Potassium, which is a +1. Calcium, +2. Magnesium, +2. Okay, chloride ions, -1. Good. Nutrients, so think glucose when you think nutrients, always think glucose. Waste products like urea or creatinine. Gases, can you give me a gas? Good. Oxygen or CO2, and different types of regulatory substances, okay, or hormones. And then proteins. So 7% of plasma is proteins. The most abundant protein in your plasma is albumins, 58%. Then globulins. Think of antibodies as one type of globulin. So antibodies are gamma globulins. And then fibrinogen. Fibrinogen is ultimately going to make fibrin for blood clotting. So albumins, most abundant. What is the role of albumins to maintain osmotic pressure of the blood? What is the osmotic pressure of the blood, guys? 0.9%. Okay, we'll talk more about that in a second. Globulins are enzymes, and some globulins transport protein sorry, globulins are, as a category, some of them are enzymes, some of them are transport proteins. I couldn't understand my slide. Sorry, guys. They transport lipids, cholesterol, fat-soluble vitamins, right? Like vitamin D, like when you're talking about alpha and beta globulins, antibodies are gamma globulins, right? And then fibrogen deals with clotting. And then 1 to 2% of inorganic salts, et cetera, et cetera. Excuse me, I want to take a second to talk about albumins. They're made in the liver from dietary protein. They make up about 60% of the protein in blood. So the majority of the proteins in blood are albumins. Second to that would be your lipoproteins, right? Like your LDL, HDL, etc. They are negatively charged and they help in acid-base balance. Because they're negatively charged, they can grab free H plus or free protons so that the protons aren't running around dropping the pH or making the blood acidic. Secondary, they transport materials in the blood that are not water soluble, like fatty acids, thyroid hormones, steroid hormones, and lipid soluble vitamins and drugs. They also transport some positive ions, kind of weird. You want to think of positive ions or negative ions as water soluble, and they are, so they're going to be dissolved in the bloodstream. But albumins will grab stuff that's not water soluble, but in addition, some positive ions. I want to focus on the drugs. So I want to tell you a little story. My father takes anti-seizure medication. He takes Dilantin, and Dilantin is transported on albumins to get from the liver, from digested material into his cells, right? Or his blood-brain barrier. So one time he had cellulitis, and the bacterial infection was under his skin, eating away at his skin, right? He had a skin wound. And he was in the hospital, and then he was transferred to the nursing home to take care of that flesh wound. And at the nursing home, the doctor called me and said his albumin levels are low, his Dilantin levels are low. And I asked the doctor, well, are we going to increase Dilantin? And he said, no, we're going to wait a little bit because his albumin levels are low, because he's wound healing. And as the wound heals and albumins creep up, his Dilantin levels should creep up as well. And that's exactly what happened. So 2 things here. When you are wound healing, whether it's a surgery or infection or what have you, you need excess proteins. Why do you need excess proteins? Because you need those amino acids to make more proteins, your structural proteins, to get that plasma membrane built. You know, it takes ATP, takes energy, takes amino acids. You need that nitrogen as well. And those fats. And so when you are wound healing and you're not getting protein and fats in your diet, the good fats, you're going to pull it from your body. And so in this case, he was pulling albumins for wound healing. His body wasn't able to make enough albumins because he was, expending so much energy wound healing. And so the liver is an amazing organ. We don't cover the liver as one lecture, but it has multitudes of enzymes in it. It does multitudes of functions. But if that liver is damaged, it's not going to make as many albumins as well. So let's say somebody has hepatitis B and that attacks the liver, you're going to see a decrease in albumins. Or if somebody has liver cancer or cirrhosis of the liver from, alcoholism or fatty liver, you're going to see a decrease in albumins. Okay. The last thing is that they create an osmotic force, a colloid osmotic pressure that maintains fluid volume within the extracellular fluid. So your plasma and interstitium, not inside the cell, intracellular fluid. Okay, take a second to wrap your brain around that. In lab 2, you looked at a colloid, a crystalloid, and a suspensoid. The colloid that you looked at was starch. okay? And that tells you the size of the particle, right? So a colloid is in the bloodstream, so it's dissolved in the bloodstream, but it can't filter through the capillaries, right? And so albumin is an example of those. So albumins are stuck in the blood. They can't get out of the capillaries because they're too big, they're colloid. And they maintain the osmotic pressure of the blood. So when I say osmotic pressure, I want you to think of particles. So osmotic pressure, think of particles. So albumins maintain the particle concentration of blood. All right, now I'm going to switch to my stylus. Hold on a second. Really? I'm just going to use a trackpad. You think this would be easier? Okay, so what is the concentration of your red blood cells? Okay, concentration of your red blood cells, so here's my red blood cell, right, is 0.9%. What is the concentration outside in your plasma? Okay, what maintains the outside concentration? Albumins do. Okay, so they're large plasma proteins. They're going to maintain the concentration of 0.9% in the plasma. So that fluid stays inside the plasma in the interstitium. The cell is actually going to regulate 0.9% inside that cell. So intracellularly. Okay, good. Let's see, globulins. So globulins transport insoluble or very small molecules. They make up about 35% of the protein in plasma. Antibodies are an example of globulins. If globulin levels are low, it can usually indicates an antibody deficiency. Other globulins, alpha beta globulins made in liver, include transport proteins like your HDL, high density lipoproteins, low density lipoproteins, very low density lipoproteins, right? And enzymes. And then fibrinogen, we're going to talk about fibrinogen. It is involved in blood clotting. All right, so let's talk about red blood cells. They're called erythrocytes, right? Erythro is red, cyte is cell. Most abundant blood cell. They're biconcave. They're shaped like lifesavers. Why are they shaped like lifesavers? Okay, so they are shaped like lifesavers because they're anucleate when they're mature, so they don't have a nucleus, so that gives them their biconcave shape. What is the purpose of that? It's to increase surface area to internal bulk. So it helps with diffusion. Anytime you're increasing surface area to internal bulk or internal volume, that is going to help with diffusion immensely. More surface area, more diffusion can occur. Anytime you see folds, that is increase in surface area. So you're gonna have more diffusion. Just like think of a baby, right? A baby has more surface area compared to bulk than an adult. So they have a harder time regulating their body temperature. They get colder easier, they get hotter easier, right? To dehydrate faster. Red blood cells live 120 days. So 120 divided by 30 days in a month, that gives you 4 months, right? So they live about 4 months. So that's why when they are looking at somebody who has diabetes mellitus, yes, they're looking at, okay, what is your blood glucose today? But they're also going to look at a record of blood glucose in your bloodstream for 3 to 4 months. When was it high? When blood sugar is high, the glucose enters the red blood cell and becomes glycosylated. It becomes stuck there. And you can take a sample of the red blood cells and measure the A1C or the glycosylated glucose and see how high the blood sugar was from the past 4 months. And so you can look at the red blood cells and make a judgment call of what's been going on in the blood sugar for the past 4 months, okay? This is another thing where you see this, or where this is important, would be sickle cell anemia. So somebody who has a mutated red blood cell because of a genetic defect, and we'll talk about that in a second, Um, those red blood cells, when you get new red blood cells, a massive amount of new red blood cells in 4 months, that person's going to be in a lot of pain, or that could be a crisis issue for the person. And then once those red blood cells, those damaged red blood cells get broken down, then they're sort of normal for a little bit. They're severely anemic, in a lot of pain still. Probably had to have blood transfusions depending on how severe it is. But you need to be aware that it's 4 months. Here's a blood smear. So you can see the red blood cells. The ones that aren't biconcave, like, let me see. This one probably up here, that's probably a white blood cell. These little dots are platelets. Here's a scanning electron micrograph. Here's what it looks like. So notice in the middle, it is 0.45. It's about 1 micrometer wide. At the end, it's about 2 to 3 micrometers, and it's about 7 to 8 micrometers long. Why is it so tiny? Because it has to fit through capillary single file, and it has to be flexible, all right? Hemoglobin is the red pigment that comprises a red blood cell. Hemoglobin is produced in the red bone marrow of long bones. There are 1 million hemoglobin per red blood cell, right? Pretty crazy. And so if you think about it, we have 1 million hemoglobin molecules per red blood cell. And then we have about 5 million red blood cells per cubic millimeter. So we have a lot of hemoglobin, and each hemoglobin can actually bind 4 oxygens. So hemoglobin is made up of 4 subunits. Each contains a heme group and an iron. That should be Fe+2. Each heme can bind to 1 oxygen molecule. So each hemoglobin carries 4 oxygen molecules, okay? Take a look at your hemoglobin. So we talked about hemoglobin before when we talked about proteins and protein folding. So here is one, so this whole thing is a hemoglobin molecule, right? This is 1 subunit. Here's another one, and another one. So it has 4 subunits, right? This is an alpha chain, this is a beta chain, an alpha and a beta. So this is alpha subunit, beta subunit, alpha subunit, and beta subunit. Because it's made up of alpha and beta subunits, it is adult. Fetal hemoglobin is alpha and gamma subunits. Adult is alpha and beta. Take a look at the hemes that are in the middle. So the hemes look like this, and then they have an iron that actually binds the oxygen, okay? So this, this iron can bind a molecule of oxygen. This can bind a molecule, this can bind a molecule, and this can bind a molecule. So we can bind 4 molecules of oxygen per hemoglobin, and we have 1 million of these per red blood cell, and we have 5 million red blood cells per cubic millimeter, okay? Here are normal red blood cells. Here is a red blood cell that the person has sickle cell anemia. So let me explain this. Sickle cell anemia is a defect in the heme form. More specifically, the alpha subunits are mutated. It's inherited, so it passes down from generation to generation, right? It's inherited from your parents. It's a defect, it's a mutation in the alpha subunit, and because the alpha subunits are mutated and they're stuck in this bag of a red blood cell, the red blood cell is going to be misshapen or sickle shaped. What was the purpose of this? This mutation occurred in Africa near the equator, right? And the purpose, well, not really the purpose of this, but this mutation occurred in Africa. People who had this mutation lived longer than people who didn't. Let me explain the malaria aspect of this. So when people are bit by the mosquito transmits the protozoan that causes malaria. The protozoans will go into the bloodstream, and they'll try to get into the sickle-shaped cell, and they won't be able to because of the sickle shape. The mosquito bites somebody who doesn't have the mutation. The protozoan that causes malaria goes into the bloodstream, and it will go into the red blood cells and hide in the red blood cells. So the immune system can't attack it. And it'll reproduce, and then it'll burst out of the red blood cells and go through the blood and wreak havoc, and then hide in the red blood cells so your immune system can't get them. And so this person who has normal blood doesn't have a long lifespan because they die early. They don't get to reproduce, right? And the name of the game for biology and evolution is reproduction, right? So if they are afflicted with malaria, they die early and they don't reproduce. The people with the sickle cell mutation, sickle cell anemia has a shorter lifespan than somebody who's healthy, who doesn't have to worry about malaria. But in this environment, this was advantageous because the protozoan couldn't get into the red blood cell, and therefore this person didn't have malaria and didn't die from malaria. And so, yes, sickle cell anemia is extremely painful, and we'll talk about that in a second. But these people were able to reproduce, and that's the name of the game. And yes, their children inherited the sickle cell mutation, but that was also beneficial in this environment. Remember, evolution is about mutations that are advantageous in that environment. So people who the people who developed this mutation were able to survive in this malaria afflicted region. And now you see sickle cell anemia in people all over the world because people travel and mate and pass on their genes. Another thing, so sickle cell anemia, because of the sickle cell shape, this can't transport oxygen very well, right? It has a lower surface area to internal volume. It's extremely painful to get this through the capillaries, and these break really easily. They're really fragile. And so a person who has sickle cell anemia, and there's different severities, you can have one gene, there's actually two genes that cause sickle cell anemia. So you could have one copy of one of the genes, or two copies of one of the genes, or one copy, you could have one of the genes and then a copy of the other one, or both copies of the other ones, or both copies of two genes, and you have a really severe form of it. So these are extremely painful to fit through the capillaries. Every 4 months, you get new sickle cell shaped red blood cells, and that is called a crisis because it's extremely painful. People, have a lower life expectancy with this, depending on how severe their sickle cell anemia is. Another thing too, because it originated in Africa, it's more prevalent in Blacks, right? And people in the tropics along the equator. Locally, it usually presents, an adolescent or puberty, when everything's revved up. A young Black girl will come into the ER and, say, I'm in severe pain. I don't understand what's going on. They'll do their basic checks. They won't catch the sickle cell anemia. They'll give her some drugs and send her away. Then in 3 to 4 months, she comes back saying, something's really wrong. I know something's wrong. They'll check her over again and think that she's just drug seeking and, give her drugs or tell her that there's nothing they can do. Meanwhile, they didn't check her blood under the microscope to see that she had sickle cell anemia. And so it's really important from a public health standpoint that your local population and what issues are in the local population, right? So for example, sickle cell anemia affects primarily Blacks from Africa in the tropical region. And it usually presents, or people start showing symptoms. They can when they're children, but usually it's, adolescent to 18 years old. If you think about other diseases, for example, testicular cancer presents in, age 25 to 35 for men, It affects Blacks and Hispanics more so than white men. And so men at that age range should be doing their testicle checks. Breast cancer, you think of breast cancer for women as age, 40 to 50 or menopause, right? But breast cancer can show in age 20, for women nowadays. It's not as unusual as it used to be. Testicular cancer is more prevalent in men in age 55, right? Think about heart disease. Heart disease is different symptoms for a heart attack in men versus women. Different drugs affect different genders and different ethnicities differently. So most drugs are tested on white men, and they don't have the same effect on women typically. And there are some cardiovascular drugs that we use, and I can't think of the name right now, but there are cardiovascular drugs that we use, and they work really well for white men. But for Black women, they can be damaging, and they don't work well. So you need to make sure that you're researching from a public health standpoint, what is the efficacy of these drugs in the different ethnicities, what are different diseases and frequencies of those diseases and different ethnicities and genders, because that plays a role in how you take care of your patients. Okay, so fetal hemoglobin, we talked about this. Elephant gamma subunits, different form of hemoglobin than found in adults, has a higher affinity for oxygen than mom's hemoglobin, and so it's going to pull the oxygen from the placenta from the mom's side. When the fetus is born, those red blood cells get destroyed because you don't need the gamma subunits anymore, and they get replaced with adult red blood cells. So you have a lot of red blood cell destruction when the fetus is born, and that can cause the fetus to be jaundiced, and we'll talk about jaundice in a second. That also gives the babies a lovely poop color because hemoglobin is recycled as bile, or the heme subunits are recycled as bile and go through the intestine, and that gives our poop its lovely brown color as it oxidizes through the intestine. And so newborns, their poop is that black sort of tar that's from all the hemoglobin breakdown that's occurring, and then it turns green as the hemoglobin, the heme stays in the bile a little bit longer, et cetera, et cetera. So that accounts for the colors that you're seeing in the colors that you see in the newborn poop is attributed to the hemoglobin degradation. And we'll talk about that a little bit later when we get into red blood cell cycling. I talk too much. Okay, so hemoglobin saturation curve. Let's talk about this. This is your hemoglobin saturation curve, and it shows positive cooperative binding. What the heck does this mean? It means when, let's back up to this picture. So what is positive cooperative binding? When oxygen binds to this, the alpha subunit, it takes a little bit of oxygen in the environment to bind it to this subunit. But once oxygen binds to this subunit, because this is a three-dimensional protein, all these other subunits snap to shape and allow oxygen to bind. So it's kind of like dominoes. You need some oxygen in this local area to get oxygen to bind to hemoglobin, and then oxygen jumps on, and oxygen jumps on. That's called positive cooperative binding. One oxygen bound causes another oxygen to jump on until all four oxygen molecules are bound, and this gives it a sigmoidal curve. So take a look at your curve. Notice this is pressure of oxygen. So it's in millimeters of mercury, right? So PO2, pressure of oxygen in millimeters of mercury. This is oxyhemoglobin, or percent saturation of hemoglobin with oxygen, right? So this x-axis is dependent or independent variable. It's the one that you're changing, so it's the independent variable, right? This one, oxyhemoglobin, or percent saturation of hemoglobin with oxygen, is the one that you're measuring, right? So is it the dependent or independent? It's the dependent. Good. So independent's always on X, dependent is always on Y. Why is it a scatter plot or a line graph? Because it's continuous data. Good. All right. So take a look at the sigmoidal curve. This is telling me that there has to be some oxygen onloading or some oxygen increase in the area before that first hemoglobin binds oxygen. Then the second, third, and fourth one bind oxygen, and then they're saturated. Okay. Keep in mind, this is not a chart or a graph of one hemoglobin. It's actually millions of hemoglobin, right? So it's showing them all sort of happening. Look at right shift and left shift after you've gone through all this material and you think you understand it, and see if you can sort of quiz yourself. I'm not going to ask you right shift, but I will ask you under which situations does hemoglobin not want to bind oxygen? So that would be a reduced infinity. Hemoglobin not want to bind oxygen. When you have a high temperature, when you have high CO2, which makes a lot of protons, and this is actually a glycolysis intermediate, which is increased 2,3, I think it's diphosphoglycerate, if I remember correctly. I know that you covered this in biochemistry. It's not something you would have covered in general chem because you don't talk about metabolism. A left shift would be increased infinity, right? So hemoglobin has a higher affinity or likes oxygen more. That would be when temperature is lower, when you have less CO2, so less protons or less acid, and less of a metabolic intermediate. Okay, so see if you can rationalize that or think through that. This is a really tough concept, but if you understand this now, it is going to serve you well in your professional programs. And we'll talk about it, of course. Okay, so I guess we're going to talk about it now. No, I'm just kidding. So let's take a look. So 50% hemoglobin saturated with oxygen. Why did I pick that number? Just because it's easy to see the difference. Okay, so at 50%, you want 50% of the oxygen of the hemoglobin bound to oxygen. How much oxygen do you need in that area? Okay, so 50%, draw a line to your curve and then down. So 30 units, right? So we need 30 units of oxygen to get 50% of the hemoglobin saturated. We need 90 units of oxygen to get the hemoglobin about 95, 96, 97% saturated. Okay, this is what normal saturation level is in our bloodstream. So when we talk about percent saturation or oxyhemoglobin, our oxygen saturation is about 97 to 100%. Okay, all right. Red blood cells, the binding of oxygen to hemoglobin occurs only when there are high levels of oxygen in a pH that is not very low, right? When oxygen is bound to hemoglobin, we call it oxyhemoglobin, and it's a bright color. At that point, red blood cells release CO2. Where does this occur? Right, so go ahead and read it again. I'll wait. Where is there a high level of oxygen? I'll give you two choices. Lungs or tissues? Lungs or tissues? Okay, so we have a high level of oxygen at the lungs, right? And you don't know that yet, but a pH that's not very low. Okay, so red blood cells are going to release CO2 at the lungs and pick up oxygen. Now you may say, okay, well, I already knew that. Yes, you did, but now you have another level to that, right? When pH levels are low, as in metabolically active tissues, hemoglobin releases oxygen, and the purple colored molecule is called the oxyhemoglobin. After releasing oxygen, the heme groups will bind CO2. Okay, so let's talk about hemoglobin for a second. Hemoglobin, and you should write this down because it's important. Hemoglobin prefers to bind carbon monoxide and carbon dioxide over oxygen. So if you have all three gases dissolved in the bloodstream, which we do, hemoglobin is going to bind CO2 over oxygen. Okay. How do we get the hemoglobin to bind oxygen? Oxygen has to be in excess, or there has to be a shift in pH or temperature. Okay, how do we get the red blood cell to bind CO2? CO2 has to be equal to or slightly higher, or equal to or slightly lower or higher than oxygen for the red blood cell to bind it, or there's a shift in temperature or pH. Okay. Okay, so let's look at the effect of temperature on hemoglobin saturation. So again, we're going to pick 50% because it's easy to see the differences. Look at the colored lines. 38, so sorry, the orange, I get excited when I talk about blood. The orange line is 38 degrees Celsius. That is our body temperature, right? Okay, so that's normal body temperature. The red line is when body temperature is elevated. The purple line is when body temperature is lower. And then the blue line is severely low, okay? So I picked 50% because that's where you can see a big difference in all of these. So 50% saturation, how much oxygen would we need in the bloodstream to get 50% of the hemoglobin saturated at normal body temperature? Okay, so that would be what? About, let's see, 30 maybe, right? Let's do the pen. So 30 millimeters of mercury of oxygen, or we can just say 30 units, okay? Okay, so at body temperature, we need 30 units of oxygen to get 50% saturation. You're following me so far? What happens if I crank up the temperature, right? So if we take this 50% saturation, right, and draw a line to our higher body temperature, if we have a fever, how much oxygen do we need? Do we need more or less oxygen to get the same level of saturation? We need more, right? So when we have a higher temperature, we need more O2 to get same saturation, right? Right? Kind of crazy, right? So, and you know this, what happens when you have a fever? What happens to respiratory rate? It's going to go up. What happens to heart rate? It's going to go up because you're sick. You're trying to get more oxygen because you're trying to saturate those red blood cells because it's harder to grab that. Okay. Let's talk about when you're colder, 20 degrees Celsius. So let's say you're at 20 degrees Celsius. How much oxygen do you need to get 50% saturation? More or less? Oops. Good. Less. Okay. So what would you call that? About 15, right? So in a colder environment, I need less oxygen to get the same level of saturation compared to normal body temperature. And I need way more oxygen when I have a fever or when the environment's warmer. You can look at this from a standpoint of hemoglobin, and biochemically how hemoglobin behaves in various temperatures, and that's what this is doing. Or you can look at it in the body and say, okay, when the body has a fever, I'm going to need way more oxygen to get the normal saturation. When the body is in a cold environment, let's say going through hypothermia, I'm going to need way less oxygen to get the same level of saturation. Okay. So as temperature increases, what happens to the affinity of hemoglobin for oxygen? Okay, what does affinity mean? Because I get that question a lot. Likes, right? So as temperature increases, what happens to hemoglobin binding oxygen? Or does hemoglobin like to bind oxygen or not like to bind oxygen? Okay, so answer that. As temperature goes up, what happens to hemoglobin wanting to bind to oxygen? Good, it decreases, right? And, oh, uh-oh. I always hate this when I Google this. Sorry, you guys. Okay, so it decreases. That looks wonderful. Normally in the body, where does this occur? Okay, so where do you have a higher temperature? At the lungs or at the tissue? Right? You're going through metabolism, you're breaking bonds, so temperature is going to go up. As temperature decreases, what happens to the affinity of hemoglobin for oxygen? So as temperature drops, what happens to hemoglobin being able to bind oxygen? It's going to increase. Okay. And where does this occur? Where is there a lower temperature? Tissues or lungs? Good job at the lungs. Okay. So yes, you kind of just rattled this off and went through this, but I want you to really understand it. I want you to draw it out. Really important. Okay. You need to start developing those drawing and critical thinking skills. Okay. The Bohr effect. The Bohr effect is the effect of pH on hemoglobin saturation. The effect pH has on hemoglobin saturation curve because of CO2. So when there is excess CO2, we have excess protons, okay? So if I took a straw and exhaled out air into my water, if I bubbled, made bubbles like kids do, I would be exhaling CO2 into the water, and that would create an acid. I could use a pH indicator and time how long it took to turn acidic, right? You can do this with your kids at home. You can look up, let me see, I think it's a cabbage pH indicator. I can't remember if it's cabbage or beet. I think it's cabbage. But you can have them, you can make a little cabbage indicator, pH indicator at home, and put it in a glass of water, have your kids blow bubbles and time how long it takes to turn acidic, and then have them run around the block and then come back and blow and see how long it takes to change. And then, you can do all sorts of stuff. You can have them run around the house. You can have them race and see who can change the water acidic faster. So it's a lot of fun to do. So you can do some chemistry at home. But anyway, okay. So what does this mean? Anytime you have excess CO2, right? So excess CO2 in water, that is going to make carbonic acid, right? This reaction is going to occur whether you have enzymes or not. However, there is an enzyme on your red blood cell that makes this occur faster, much faster. It's called carbonic anhydrase. So that's on your red blood cell. So excess CO2 plus water in the presence of carbonic anhydrase is going to make carbonic acid. Okay, here's the reactants, here's the products. When you have the carbonic acid, as soon as you have it, it's going to dissociate into a proton and bicarbonate ions. Protons, what do protons do? Are they acidic or basic? They're acidic, right? So when you have an accumulation of protons, what is that going to do? It's going to make it more acidic, right? And what does that do to pH? Acidic, is it a lower pH or a higher pH? It's a lower pH, okay? Yes, you're going to make bicarbonate ions too. When we get into respiration, we'll talk about this a little bit more. The protons actually stay stuck to the red blood cells. The bicarbonate goes out in the plasma. So notice bicarbonate is a negative. This is actually one of our major buffering systems in our blood as well. The ocean has bicarbonate as well. So the goal of bicarbonate, it's HCO3 negative. We'll throw some albumins in there, right? Just so you guys get it. When you make a lot of CO2, like you're going through metabolism at the site of the tissues, you're going to have a lot of H+, right? H+ or free H+ floating around is dangerous because it's going to drop the pH. It's going to make it more acidic. So what do these albumins and bicarbonate do? They bind the H plus because they're negatively charged, right? They bind the H plus and take them out of solution so they're not dropping the pH, right? What drops the pH of the blood? Free H plus. When it's bound to albumins and bicarb, it doesn't shift the pH of the blood. And then the kidneys are going to kick out the H plus, and your lungs are going to deal with the CO2. All right, so the effect of pH on hemoglobin saturation. What is normal blood pH, guys? 7.35, 7.45. Good, so 50% saturation, right? If we go to normal blood pH, we need about 30 units. Notice how the numbers are relatively the same. If we are in a more acidic environment, right, do we need more or less oxygen to get the same level of saturation? We need more, okay? So if you drop pH, do you need more or less oxygen to get the same saturation? You need more O2 to get hemoglobin saturated, okay? What happens when you increase the pH to 7.6? Oops, sorry. You need less oxygen, right? So about 18. So normal pH, you need about 30 units of oxygen. When the pH is acidic, you need more oxygen to get the same level of saturation. When the pH is more basic or less acidic, you need less oxygen. Okay, so as pH decreases, what happens to the affinity of hemoglobin for oxygen? It's going to drop, right? So hemoglobin doesn't want to bind oxygen when when it's an acidic environment. Where does this occur? Lungs or tissue normally. Good. Okay, so take a look at this. Here's your hemoglobin saturation curve. When you have high oxygen, a high pH at the lungs, hemoglobin binds oxygen easier. When tissues produce excess CO2, low pH, right? Hemoglobin is going to release oxygen easier. So when there's high oxygen, hemoglobin binds oxygen easier. When tissues produce excess CO2 at low pH, hemoglobin is going to release oxygen better. Okay, so if you take a look, here's your hemoglobin saturation curve. The black line is normal, right? Black line is normal. The blue line is what? So without even looking at it, if you could cover this up, you can figure it out. So if we get 50% saturation, in order to get 50% saturation, you need more oxygen. So that would be in a hot environment or high CO2 environment, right? And so if you look at this, hot, acidic, high CO2 environment. Okay. In order to get 50% saturation, with the red line, you need less oxygen. So what type of an environment is that? It is a low CO2 environment, right? Or a cold, basic environment. Good. And then this is obviously myoglobin. Okay, so I'm going to leave you with this as sort of a little homework assignment. On the left side is the tissues. On the right side is the lungs. So I want you to walk through this. Lungs is our high oxygen, low oxygen, high CO2, low CO2, and CO2 is going to dictate the protons. Protons is going to dictate pH and then temperature. And then do the same thing for the tissues. And then I want to know, in between this is the blood, right? So I want to know when the red blood cell comes over to this area, which gas is it going to grab and why? What are the parameters? All right, when the red blood cell gets to the tissues, which gas is it going to grab and why? Okay, So first, your rules are the red blood cell is going to grab whatever gas is in excess, right? Keep in mind it has a higher affinity for CO2 than O2. And then after that, pH and temperature are going to have an effect as well. Okay, so we're going to stop there. We'll pick this up next lecture. So make sure you draw that out.